Our next student, I just learned, is actually a recent graduate of the University of Michigan as an undergraduate and has been working with Ruya Pakkan and Kostas Lysiotis. And Ruya Pakkan will be talking about employing mass spec to characterize the metabolic adaptations of PIK5 inhibition in pancreatic cancer.
My name is Ruya, and I'm going to talk about how we use mass spec to characterize the metabolic adaptations to PIK5 inhibition in pancreatic cancer. So, some background: pancreatic ductal adenocarcinoma, also known as PDAC, is one of the deadliest cancers. It has a survival rate, 5-year survival rate of around 13%, and right now the therapeutic options they have are very minimal. This is because PDAC cells, the microenvironment they're in has a very high degree of stromal fibroblasts and extracellular matrix deposition. This makes PDAC cells very good at thriving in high-stress environments, and they can use different intra and extracellular pathways to scavenge for nutrients. A lot of groups are trying to target these pathways that PDAC uses to scavenge nutrients in order for therapeutic options. Most recent studies have been looking at the autophagy pathway and trying to target autophagy or the lysosome in order for a therapeutic strategy, but there aren't very good points of inhibition in this pathway currently to target, and there hasn't been a lot of success. So my group that I've been working with has been looking at how to target autophagy in novel ways in order to target PDAC cells. So what we ended up looking at was PIK5, which is a lipid kinase, and PIK5 is used in the body to convert PI3P into PI3-5P2, which is really essential for lysosomal functioning. So when we thought that if we inhibited PIK5, we might be able to target autophagy in the lysosome in a different way than other lysosomal inhibitors have been targeting. So that was the goal of our project. And what we did initially was to make sure that PIK5 perturbation does suppress autophagy and see what happens to PDAC cells upon PIK5 perturbation. So we tried this in 2 different methods. First, we used apilamod and ESK, which are both phase 1 cleared inhibitors of PIK5. They've been tested with different cancers in phase 2 currently. So we performed a Western blot on 7940B cells, which is a mouse PDAC cell line, using apilamod and ESK. And in both cases, P62 and LC3 are markers of autophagy. And we can see that using apilamod and ESK, we successfully inhibited autophagy upon PIK5 inhibition. We also tried another method using CRISPR interference, where we knocked down PIK5 from Ruya Pakkan 2, which is a human PDAC cell line. And as you can see from the first line here, we successfully did knock down PIK5 from these cells. And then again, we used P62 and LC3 to make sure that autophagy is in fact inhibited when PIK5 is knocked down from cells, which it was. So then we looked further into these cell lines that we knocked out PIK5 from, and we compared them to the normal cell lines. And here we can see that we have a growth curve. The confluence, so the cell growth was a lot slower when PIK5 was knocked down from the Ruya Pakkan 2 cells compared to cells that do have PIK5. So after we discovered this, we, I guess, had 2 main questions. The first thing that we were thinking about was we were trying to figure out if these cells were growing slower because upon PIK5 inhibition, we discovered autophagy is being inhibited. So we were trying to see if the reason these cells were growing slower was because autophagy is being inhibited upon PIK5 inhibition, or if PIK5 had a different original role in these cells that is making them grow a lot slower. And then the other question we posed was yes, these cells do grow a lot slower when PIK5 is inhibited, but they're still growing. So we wanted to see what these cells were relying on upon PIK5 inhibition in order to grow. So, as a lot of biologists do, after we posed these questions, we did a CRISPR screen to understand the situation a little better. And we used the Ruya Pakkan 2 cells again. We used a library with 30,000 guide RNAs and 3,000 genes with around 10 guide RNAs per gene. And we used apilamod because it works better in vitro with DMSO, and carried out our CRISPR screen for 14 days and then got our results sequenced to see. And what we found from our CRISPR screen was that almost all of our top hits were fatty acid synthesis genes. As you can see here, some of them are ACACA, FASN, HSD17B12, and ELOVL1. So this kind of told us that upon PIK5 inhibition, the cells rely on fatty acid synthesis in order to keep growing and keep proliferating. So we kind of thought that PIK5 had this function in these cells that yes, it does block autophagy, but it also had another function in these cells that fatty acid synthesis was getting, that the cells were relying to fatty acid synthesis to compensate after PIK5 inhibition. So then we did an RNA sequencing experiment with apilmod and ESK, and we did an 8-hour treatment, and we found that after 8 hours of treatment with apilmod and ESK, cholesterol homeostasis, mTOR signaling, and fatty acid metabolism were the most upregulated pathways. And here we have a volcano plot showing our most upregulated genes. And again, these are all like fatty acid synthesis or cholesterol genes. So kind of going back to fatty acid synthesis thing, what we realized is not only do cells rely on fatty acid synthesis more to proliferate and live once PIK5 is inhibited, but also fatty acid synthesis is upregulated upon PIK5 inhibition. So we wanted to understand the mechanism of how PIK5 inhibition is causing fatty acid synthesis to go up. So we looked at SRBP1. SRBP1 is one of the key regulators of lipogenesis. So we first looked if SRBP1 gets activated upon PIK5 inhibition. So we again used apilmod and ESK, and here's the premature bands of SRBP1, and here there's we can see the mature band with apilmod and ESK treatments. So we confirmed that SRBP1 does get activated upon PIK5 inhibition. We then wanted to use phaedostatin to see how this relates to fatty acid synthesis. So phaedostatin is an SRBP1 inhibitor. And here with phaedostatin, we successfully blocked SRBP1 from being activated even upon PIK5 inhibition, even though it normally does. Then we ran a quantitative PCR using FASN, which is one of our fatty acid synthesis genes. And here we can see that without phaedostatin, upon PIK5 inhibition compared to DMSO and apilmod, apilmod ESK compared to DMSO, FASN transcript levels are going up compared to DMSO significantly, which is what we expected from our RNA sequencing data, because we did discover that fatty acid synthesis goes up. So then we added phaedostatin, and when we layered this with apilmod and ESK, yes, FASN transcription levels did go up still, but it was not as significant as without phaedostatin. So we did determine that to some extent, fatty acid synthesis is going up through SRBP1 activation because phaedostatin inhibits SRBP1. So that's kind of the mechanism that we drew. So now getting to, I guess, the actual analytical chemistry part of things, we wanted to see how the lipid metabolism is affected with PIK5 inhibition. So we performed a lipidomics experiment with Agilent, and Pietro Morlacci performed this in Massachusetts, Lexington, if I'm not mistaken. And what we did was we took our cells, we treated them with our PIK5 inhibitors, and then we gave them to Agilent. And they were able to use their Bravo liquid handler in order to separate the polar metabolites and our non-polar metabolites, lipids that we're interested in. And then they used LC-MS-MS to give us kind of a profile of what lipids were how much upregulated and downregulated in the cells after PIK5 inhibition. And what we observed was a very large increase in sphingolipids after 24 hours of PIK5 inhibition. Here, specifically, hexacylceramides, sphingomyelin, and ceramides were upregulated. This is compared to DMSO, this vertical line here, with apilmod and ESK. Another thing to note here is that in all of these conditions, we can see that the apilmod and ESK lines, in this forest plot, are lying very close together, even though these are two independent, separate PIK5 inhibitors. So we confirmed that this trend we observed with lipids is in fact due to PIK5 inhibition and not something else going on in these cells because of these inhibitors, but it is because of PIK5 inhibition. So then we looked at sphingolipids further, and we made these volcano plots. And for both apilmod and ESK, we can see that a lot more sphingolipids are upregulated than downregulated with PIK5 inhibition, so they become the most significant group of lipids that get upregulated. So then we did another targeted lipidomics experiment looking at SRBP1 again, because we had determined that it related to the fatty acid synthesis pathway. We wanted to see if SRBP1 activation also affected lipid synthesis on like metabolism level. And here we have a PCA plot. Here's our DMSO condition represented in blue. And then here in red, we have apilamod and ESK. And as you can see, like there's some overlap in apilamod and ESK, and they're further away from DMSO. What we wanted to see was if we could kind of rescue the kind of pushback we get from apilamod and ESK with phaedostatin. So if the like lipid levels would like get closer to DMSO when we blocked SRBP1, because that would show us that sphingolipid synthesis does increase with SRBP1 activation, and it is partly responsible for it, which is exactly what happened. As you can see here, we have apilamod, and then here we have apilamod and phaedostatin together. Gets a lot closer to DMSO, and same with ESK. We see the same trend here. So at least partly, SRBP1 activation does cause an increase in sphingolipid synthesis. And we have some violin plots here for ceramides and hexosylceramides. And again, we can see that with apilamod and ESK, when we layer phaedostatin over them, we get some rescue back to the DMSO condition. So going back to this pathway, we kind of wanted to see where, and why these sphingolipid synthesis was getting replicated, and I guess where they were coming from. So our attention got drawn to another gene that was one of our top 10 hits in our CRISPR screen, which is SLC25A1. That's a citrate transporter that transports citrate from the transport citrate. And we hypothesized that maybe some glycolytic intermediates were getting converted into citrate upon PIK5 inhibition, which was then getting shunted into de novo lipid synthesis. To test this, another thing that alerted us for this was that we performed a metabolomics experiment that I don't really have time to talk about, but we saw that after 3 hours of treatment, citrate was going down, and then after 8 hours, it was getting recovered, which is why we formed this hypothesis. And what we did was, to understand this, was to do stable isotope tracing, which we performed in the Lysiotis lab. We see their 7940B cells, which is our mouse cell line, mouse PDAC cell line, as we normally do, incubate them overnight. And then we changed their media that they grow in from the normal glucose media we have. We prepared a 13C glucose media and treated them with our PIK5 inhibitors, and we had a control for 24 hours, and then we performed a mass spec. And the results we got from this experiment were exactly what we expected. We can see that for C20 ceramides, which has 38 carbons, with PIK5 inhibition, the C13 incorporation is much higher than with DMSO. So with the PIK5 inhibited conditions, the cells did in fact take up more of the glucose and convert it into sphingolipid, specifically ceramide for this case, but we did it for other ones too. And then we have kind of a box plot here that shows the same thing. We can see that there's a much more of an abundance of higher weight ceramides with apilamod and ESK, so our PIK5 treated conditions compared to our DMSO condition. So some of our conclusions was that PIK5 controls lysosomal nutrient recycling and acquisition pathways in PDAC, and that PIK5 inhibition drives lipogenic transcriptional and metabolic program that results in diverting glucose to synthesize lipids, and specifically sphingolipids. And our future direction right now, what we're working on, is looking at more of the cholesterol side of things, because that was the other thing we got from RNA-seq and our CRISPR screen, where cholesterol genes and the cholesterol pathways going up, which we found also very relevant. So we're kind of repeating the same experiments for the cholesterol pathway this way and see how it plays into things. I would like to acknowledge, first of all, Caleb, my mentor. He is an MD PhD, for mentoring me for the past year. And then I'd like to acknowledge Lee and Peter for helping us with the analytical chem part of things, like in Michigan, and then Pietro and Bianca from Agilent, who helped us perform our lipidomics experiments. And then Kostas and Nurul for having me in their labs. Thank you so much.